Hello, class. My name is Professor Gates, and this is the video for Lecture 1, Introduction to Anatomy. All right, then. So, Introduction to Anatomy, it's based on Human Anatomy by Martini, Chapter 1. Your to-do list is to read the lecture, listen to the lecture, do drawing assignments and homework questions. If time permits, you can read the chapter in Martini's and you can do additional practice questions. Here are the student learning objectives. By the end, you should be able to explain the following points, be able to describe following points, be able to use anatomical terms. You can read those all. After the student learning objectives come the drawing assignments. And here is your drawing assignment 1 and 2. And then a special labeling assignment, not to be turned in, but just to learn from. And then the lecture slides are in blue. Questions, homework questions are in pink. When we get down to the end, the lab slides are in green. The additional practice questions are in dark green, and the answers to the additional practice questions, which you will not be handing in on a Scantron, are in gray. Now, this is a huge pack of slides, goes to 146 slides, but the actual information slides are about 60, and I use big writing and a picture on every slide. So it's doable. All right, so to start our lecture, Introduction to Anatomy, Structures and Functions. So anatomy is the study of the structures of the body, whereas physiology is the study of the functions of the structures of the body. FYI, the word anatomy comes from the Greek word that means to cut apart. So that's where that came from. And structures and functions of the body have evolved together over billions of years. Therefore, structures and functions of the body are very interrelated. And here's a nice picture showing how a long time ago life began. You can follow the spiral up, 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 up. And finally, we get to this little human here. Of course, that would be us. What is a structure? A structure is a complex anatomical part of a living thing. So it could be pretty much anything. A structure could be a bone, it could be a muscle, it could be a cell, it could be a tiny little organelle in a cell. So we're just going to leave that word as a kind of all-purpose word for a complex anatomical part of a living thing. A function is the activity of a structure or how it works. Specific anatomical structures in the body allow the body to perform specific functions. So, for example, this is a shoulder and an upper arm and then a lower arm shown here. And the bicep brachii muscle, which is, the muscle in the front that you show off when you want to show people how big your biceps are. Can I bring put mine in there? Ooh, yeah, it's huge. Oh my goodness. The bicep brachii muscle is attached to the scapula bone at the shoulder joint and the radius bone at the elbow joint. And its function is flexion of the elbow joint and the shoulder joint, and it has some other minor functions. And it's the structure of the bones, the muscles, more bones that allow flexion of the elbow joint and the shoulder joint to occur. Of course, we'll learn a lot about that. All right, introduction to anatomy, types of anatomy. There are multiple types of anatomy. Different types of anatomy are helpful in different clinical situations. If you're thinking of going into any kind of a clinical job, okay, whether it be a nurse, a PA, a doctor, a physical therapist, a physical trainer. Different types of anatomy are helpful in different clinical situations. First, the different types can be put into two big buckets. Gross anatomy is the study of structures that can be seen with the naked eye. Microscopic anatomy is the study of structures that are only seen with the help of magnification. So first, we'll talk about types of gross anatomy. So systemic anatomy, which is how this course is organized. Systemic anatomy is the study of organ systems, which is where the word systemic comes from. And an organ system is a group of organs that perform a function, whereas an organ is a group of cells and tissues that performs a function. And here on this slide are shown 11 different organ systems, all of which we're going to study. Not going to skip any. That's how we're going to approach anatomy. Also helpful is surface anatomy. And for surface anatomy, there are many clinical correlations. Surface anatomy is the study of how superficial anatomical markings seen on or deep to the skin relate to deeper structures. So surface anatomy is used to choose where to draw blood, for example. The median cubital vein is easily seen under the skin and is a common site for drawing blood. If you've ever drawn blood, or if you've ever had your blood drawn, you'll know that there are a lot of very visible veins in the antecubital region, or the region in front of the elbow joint. Here's another clinical correlation for surface anatomy. Surface anatomy is used to choose where to insert a needle between vertebrae to draw out some of the fluid that surrounds the spinal cord. This is called doing a lumbar puncture or spinal tap, and it's often done between L3 and L4 vertebrae. These are a combination of visible spinous processes of L3 and L4 vertebrae and palpable spinous processes of L3 and L4 vertebrae. A lumbar puncture may be used to diagnose a central nervous system infection, for example, something like meningitis. And here's another clinical correlation for surface anatomy. The sounds of the valves of the heart are best heard in locations between certain ribs. And these red dots in this picture show the locations of the places where the valves are inside the body. And in this smaller print here, there's a description of where exactly the surface markings are. So let's take the first dot. The aortic valve is heard at the right second intercostal space near the sternum. So you can palpate and to some extent on some people observe the second rib, and then below it is the second intercostal space, and also easily seen in many people and palpated is the sternum, the bone in the center of the chest. And so those surface markings can be seen and palpated and help us to know where an underlying structure like the aortic valve is, and so on for each of the valves. Okay, so those are three different ways that we can use surface anatomy in clinical work. And you may say, well, why aren't we just studying surface anatomy then? Well, it's limited. It's not like you can study the liver in depth using surface anatomy, for example. This just works for some things. Okay, so moving on. There's regional anatomy, and that is the study of a region or area of the body. For example, in the neck, parts of many organ systems are packed together in one region, the region of the neck. Regional anatomy is an advanced way to study anatomy. It's very difficult to learn it this way. That's why we learn it using systemic anatomy instead of regional anatomy. And even though it seems so natural and organic to study using regional anatomy, but it's in practice rather difficult. So it's not usually taught that way to beginners. But for example, if you were a surgeon that needed to operate on the neck, you would have to learn the regional anatomy of the neck in depth. So you'd know exactly where everything was, um, including important things to avoid, like blood vessels, which you wouldn't want to cut into. All right, developmental anatomy and embryology. Developmental anatomy is the study of the changes in anatomy from conception to physical maturity, whatever age that would be, 21 or whatever. Embryology is the study of development from conception, when an egg is first fertilized by a sperm, to 8 weeks. And a lot goes on during that time, and study of the development of an embryo is very interesting and will not be covered very much in the course. Okay, comparative anatomy is the study of the anatomy of different animals. For example, a limb with 5 digits is seen in most animals, and this shows our common evolutionary ancestry. And I have to say, no matter how many times I learn it, I find it, you know, just short of unbelievable that I had a common evolutionary ancestor with a bird, a frog, a turtle, a whale, and a horse. I mean, just mind-blowing. But such is the amazing study of evolution. Now, this picture looks very complicated, but if you read it, it's just very interesting. It's showing the basic vertebrate body plan. And here they're showing a salmon, a chicken, and a human. And they're showing that when they're embryos, uh, we actually look similar. Although, of course, by the time we're adults, we look extremely different. All right, so that was gross anatomy, the different types. So we had systemic, surface, developmental, including embryology, and comparative. Now, about microscopic anatomy. Cytology is the study of cells, and histology is the study of tissues, and a tissue is a group of cells that work together. This class studies a lot of histology, tissue, okay? However, clinically, both cytology and histology are useful. For example, to diagnose cervical precancer and cervical cancer, both cytology and histology are used. Cytology is used first. Cells are taken from the cervix and examined. And this is actually called a pap smear after a man named Papanicolaou. However, the original way that cells were examined on a smear is no longer done. They're now cells are put into a little bottle of formalin to be preserved, and then they're spread out on a slide by a machine, nice and thin, so that they can be examined easily rather than in a smear, which might have irregular layer of cells, some areas thick, some areas thin, some of the cells crumpled up, etc. Uh, this way is better, but anyway, it's still called a pap smear. Then histology is used if the cells are abnormal. A tissue biopsy, a tiny chunk of tissue, is taken from the cervix and examined. And the results of the tissue biopsy give the diagnosis. So cytology is used as a preliminary screen for anything that looks suspicious for cervical precancer or, worst case scenario, cervical cancer. And then a follow-up tissue specimen is used to make the actual diagnosis of cervical precancer or cancer. So both can be used. We'll mostly, with like a couple of exceptions, we'll mostly be looking at histology, not cytology.